translation, which is translating from the nucleotide language of RNA into the amino acid language of proteins. Transcription happens in the nucleus and is from nucleotide to nucleotide, which simply means to rewrite transcription. Translation is changing it from one language to another. In this case, we're going from RNA nucleotides to proteins, written in the amino acid language. How does mRNA code for proteins? There are only 4 letters in the code, GCAT, and those get transcribed directly into 4 letters of an mRNA code, GCAU, where U takes the place of T. This needs to be translated into proteins, and proteins have 20 different variations of amino acids. So how can you code for 20 amino acids with only 4 bases, G, C, A, and U? For a one-to-one relationship isn't going to be enough. 2 will only get us to 16. And that's obviously not enough. We need to get at least to 20. And so if we have combinations of 3 letters together, we can make a code that has more than enough for our 20 amino acids. So mRNA codes for proteins in what are called triplets, which is called a codon. And so the mRNA is what is read for the code to make the amino acid. And each sequence of 3 triplets, a codon, codes for 1 amino acid. So each one of those 3 codes for 1 amino acid, which you'll see in the preceding slides. So in this one, the code is shown by a chart, which we will be practicing. The code is for all of life, bacteria all the way to humans, and this supports what we've talked about in our evolution unit of common ancestry. All organisms use the same code. The start codon happens to be AUG, and that begins us by placing the amino acid methionine. When we get to the end, after we've translated perhaps hundreds of thousands even of the sequences to make hundreds of thousands of up to maybe perhaps even 20,000 long amino acids, we're going to get to the end, and the end is denoted by stop codons. There are 3 stop codons, UAA, UAG, and UGA, and they code for no amino acids, and when you get to the end, it simply puts in space. It terminates the sequence. Here's a couple of animations. One is by the University of Nebraska, and that is a animation that you can step through and read. And the second one is a video, which is about 4.5 minutes, that tells you about DNA translation. Can you tell the story from this picture? So from this picture, we want to start with transcription inside the nucleus. We want to go outside the nucleus in the cytoplasm and do translation. So let's begin inside the nucleus. Inside the nucleus, you have the DNA. The DNA is unwound and read in a process called transcription, which makes mRNA. The mRNA then leaves the nucleus and goes to the cytoplasm. In the cytoplasm is going to bind with the ribosome. At the ribosome, the codons, or the triplet codons, the triplet sequences will be read and translated into amino acids. These amino acids will link together in a long chain and form the protein. And this entire process is called translation.